channel. So in the previous videos, in the series of microbiology, I actually taught you about Koch's postulates and culture media. I hope you have seen those videos. If not, please go and watch it. So continuing the series of general microbiology, here is another video. In this video, we'll start with general bacteriology. But before we jump into general bacteriology, let's learn some of the name of bacteria. The basic bacterial classification is done based on gram staining. They can be either gram positive or gram negative. Whereas on the basis of shape, there can be cocci, bacilli, and comma shape, spherical, and those others. But the most important ones which we need to remember are here. I have listed them here. These questions are asked mostly in vivas. Your teacher can ask you name some gram positive cocci or bacilli, or gram negative cocci or bacilli. So let's learn them. First, looking into gram negative bacteria and gram negative cocci. So Neisseria gonorrhea and Neisseria meningitis are gram negative cocci, whereas gram negative bacilli, a very important family of bacteria, is Enterobacteriaceae, which include E. coli or Escherichia coli, Klebsiella, Enterobacter, Citrobacter, Salmonella, Proteus, and Shigella. These are all gram negative bacilli. Coming to gram positive cocci, Staphylococcus aureus, species of Streptococcus like Streptococcus aureus, Streptococcus pyogenes, Agalactiae, pneumonia. And other than that, there is Enterococcus and Viridans streptococci. These are the gram positive cocci. Coming to gram positive bacilli, it includes Corynebacterium diphtheria, Bacillus anthracis, Mycobacterium tuberculosis, Mycobacterium leprae, and Listeria. Now let's start with general microbiology. Now let's start with general bacteriology. The bacterial cell wall, which is the outermost layer of the bacteria, we'll look into it first. So this bacterial cell wall, it is a tough and rigid structure which surrounds the bacteria. If this is the bacteria, supposedly, the outer structure, which is very rough and tough, is the cell wall or the bacterial cell wall. What is the function of bacterial cell wall? Why is it present there? So 5 of the important functions are listed here. Let's go through them one by one. The bacterial cell wall, it provides protection against osmotic lysis. The bacteria are the kind of structure which actually exist in very hostile kind of environment. Like they can exist and survive in extremes of environment. So for that, to protect them against those extreme measures, the bacterial cell wall plays an important role. Like if the bacteria there are in a place, or like if they grow in water, there are chances of osmotic lysis. So the cell wall, it protects them against osmotic lysis. They act as virulence factor, and they can also act as endotoxin. Virulence factor is the factor which actually causes antigenicity in the host or bacterial host and help them to survive in the host. They provide rigidity and make it like a tough surface. It is because of the presence of peptidoglycan on the cell wall. They also provide them immunity. How? So the antibody which is produced against specific cell wall antigen, it gives immunity against some bacterial infection. So this immunity is not actually for the bacteria, but it's for the host. If the bacteria invades a host, then immunity or immune system is activated against this bacteria or the foreign substance in our body. Now, the bacterial cell wall, they actually produce cell wall specific antigen against which our body can produce antibody and help prevent the bacterial infection, like antibody to the LPS. And the fifth function is that they protect the cell or bacteria from toxic substances. Now let's see gram positive and gram negative bacterial cell wall one by one. Starting with gram positive. They're actually simpler than gram negative bacterial cell wall. So the if we talk in terms of complexity, the gram negative bacteria, they are found to be more complex than gram positive bacteria. Now we'll see what the cell wall is made up of. So the first layer here is peptidoglycan. The peptidoglycan layer in gram positive bacterial cell wall is thicker. Each layer of peptidoglycan, it is composed of mucopeptide chain, and these mucopeptide chain, they are made up of NAM and NAG molecule, an acetyluramic acid and an acetylglutamic acid, which cross-link via a tetrapeptide side chain of a pentaglycine bridge. Also, the other statement here says that the tetrapeptide chain, it ascends from NAM molecule. It is composed of L-alanine, D-glutamine, L-lysine and D-alanine. You don't really have to remember these in term, but just remember that this, like, just know about. So it will be easier for you to write the differences between gram positive and negative bacteria. Here the alanine, glutamine, lysine, alanine, these are amino acid, and DNL are their enantiomers. So the tetrapeptide chain which is present here between the cross-linking of NAM and NAG molecule, they ascend from NAM molecule and they are made up of these amino acids alanine, D-glutamine and lysine alanine. The second layer is teichoic acid. Teichoic acid is a polymer of glycerol and ribitol joined by phosphate group. So there is glycerol and there is ribitol which are joined together by phosphate group. This is the structure of teichoic acid, and not this is actually a monomer, but a polymer of this, like this structure repeating many times, makes up teichoic acid. This teichoic acid, it maintains the structure of cell wall. The two types of teichoic acid present are cell wall teichoic acid and lipoteichoic acid. Now that we have seen here, let's look into the diagram. This is the diagram which I have drawn it in a very simple manner. You can also copy this in your exams, uni and theory papers. As we'll start from here, these small round and then this lobular structure which we are seeing here, it is actually the plasma membrane. Here is the plasma membrane. These are all the phospholipids, and the purple colors which I'm indicating here, these are proteins. The intrinsic protein and extrinsic protein, you must have heard of them. These are the intrinsic proteins actually which invades into the plasma membrane. Now above it is the periplasmic space, and if we go above periplasmic space, you'll see the peptidoglycan layer. This peptidoglycan layer is actually the first layer of cell wall, and In this peptidoglycan layer, as we can see, I have tried to show the bonds here. There is presence of teichoic acid and lipoteichoic acid. So these three structure here actually constitutes the cell wall of gram-positive bacteria. Now let's go to gram-negative bacteria. This is a diagram, but let's first read about it. So gram-negative bacterial cell wall, they are thinner and more complex. They are composed of peptidoglycan layer. There is outer membrane and there is lipopolysaccharide. So it is, you can see that it's different from the gram-positive one. The peptidoglycan layer, it consists of alternate NAM and NAG molecule, but the difference is that there is no pentaglycan bridge present in gram-negative bacteria. The outer membrane, which is present outside the peptidoglycan, it consists of OMP or outer membrane protein, also known as sporin, which help in transport of molecule across the cell wall. Then there is lipopolysaccharide or LPS. It consists of three structures, that is lipid A, there is called polysaccharide, and O side chain or O antigen. Now let's look into the structure. Again, here is the plasma membrane with the phospholipid layer bilayer. Then these are integral proteins. Integral protein can be extrinsic, intrinsic, if you remember. Then above it is the periplasmic space and the peptidoglycan layer. As you can see, it is thinner than the gram-positive bacteria. Then there are some proteins here invaded in the peptidoglycan and the periplasmic space. It is known as bronze lipoprotein. Above the peptidoglycan layer exists the outer membrane. This outer membrane, it has outer membrane protein, C, also known as sporin. Then above it is the lipopolysaccharide layer. These three lipid A, these are the lipid A. Then these round structures, these are lipid A. Above it is the core polysaccharide, and above it is the O specific side chain. These three constitute the lipopolysaccharide or the LPS. Now I have summarized the difference between the cell wall of gram-positive and negative in a table here. So here are the characters. The peptidoglycan layer is thicker in gram-positive, right? So they are thinner in gram-negative. The pentaglycan bridge is present in positive but absent in negative. The lipid content you'll find nil or scanty in case of positive, but it is present in gram-negative. The lipopolysaccharide is present only in gram-negative. It also constitutes or makes up for the endotoxin of the bacteria. The teichoic acid we saw only in case of positive. Then the variety of amino acid are found in gram-negative, but it is quite less in gram-positive. So now that we are done with cell wall of bacteria, let's move into the another structure known as mesosome. Mesosomes, these are the invaginations of plasma membrane in the shape of tubercle or vesicle or lamellae. They are more prominent in case of gram-positive. So if you didn't understand, let's just assume this is the bacteria and this is the outer one is the cell wall and this is the plasma membrane. If the plasma membrane invaginates, or let me show you another one. This is the cell wall and this is the plasma membrane which invaginates the cytoplasm, and in different structure, then these invaginations are known as mesosomes. I hope you got it. Now let's look into the function of mesosome. They help in bacterial respiration. They possess respiratory enzymes which are analogous to eukaryotic mitochondria. The function of a mesosome is quite similar to the function which is performed by mitochondria in case of eukaryotes, which includes the bacterial respiration. And the second function is that it can help in cell wall formation, chromosome replication during the case of cell division or bacterial division. Now that we are done with cell wall and we are also done with this mesosome, let's look into the cell wall appendages. Cell wall appendages are, in case of bacteria, there is a special layer present outside the cell wall which is known as glycocalyx. So it is not present in all the bacteria, but some of the bacteria they possess amorphous viscid material outside the cell wall. It is known as glycocalyx. Glycocalyx, if it is well organized and it cannot be easily washed off, it is known as capsule. But if it is diffuse, unorganized, and a loosey material is there which can be removed easily, it is known as slime layer. So here is one exception. Streptococcus salivarius is a bacteria which possesses both capsule as well as slime layer of the glycocalyx. Now, what are the functions of capsule? Capsule, it contributes to the bacterial virulence. As I told you earlier, virulence is like a factor which leads to which actually help the bacteria in causing infection to the host. Those factors are the virulent factors. So the capsule is one of the virulent factor. We can say the capsule, they protect the bacteria from phagocytosis. They prevent the complement mediated bacterial cell lysis in the host cell. They prevent dehiscence, that is drying out, dehydration, these kind of cases. They may be toxic to the host cell, and they also help in biofilm formation and adhesion. So this was the first. Under the heading of bacterial virulence, we can see that capsule protect the bacteria from phagocytosis. It prevent dehiscence. It may be toxic to host, and it help in biofilm formation and adhesion. Now we'll see what is biofilm. I made it up here. See. So biofilm is actually a living ecosystem which consists of millions of bacteria which are in a self-produced matrix which is made up of extracellular polymerized substances, that is polysaccharide slime layer. So this is like a ecosystem, a small living ecosystem which is made up of millions of bacteria. And what is the environment of this ecosystem? So you'll be surprised to know that this ecosystem is produced by the bacteria themselves by this matrix which is made up of extracellular polymerized substances. They can be polysaccharide slime layers. This is biofilm, and this biofilm formation capsule is one of the factor which helps in biofilm formation. The second function of capsule is it can act as vaccine. So it is like this point is in favor of us, not bacteria actually. Now, how will you demonstrate a capsule in lab? So different ways to demonstrate capsule in a laboratory. We'll see them one by one. First is negative staining by India ink or necrosin stain. So the capsules, they appear as clear refractile halo. Can you see the halo like this pen? If I draw something or anything here over here, just see the like margin. Can you see it a little bit bright and right? And now assume that this shape or whatever I made is actually a substance, and you're looking it in under the microscope. And when we look it into through the microscope, a refractile halo is seen. Like if you change the adjustment, it will like disappear, appear, disappear, appear. That kind of appearance this capsule gives and background, and both background and bacteria, they are black. So what do we get? That bacteria and background both are black, but the capsule, it appears as a clear refractile halo. This is how we can demonstrate capsule by using negative staining. One of the ink are India ink and Negrosin stain we can use. Now the second method is McFadden capsule stain. This capsule of Bacillus anthracis by using polychrome methylene blue can be demonstrated. So we are using polychrome methylene blue stain to demonstrate the capsule of Bacillus anthracis. Third method is the serological method. The capsule material is antigenic and it can be demonstrated by specific anticapsular serum. So there is Quellen reaction where the capsular serotype of Streptococcus pneumoniae, it can be detected by antisera mixed with methylene blue. Here the capsular serotype, we are using the bacteria Streptococcus pneumoniae. We can detect the capsule of this bacteria by using antisera which is mixed with methylene blue. This is Quellen reaction. The second is capsular antigen, which can be detected in sample by using latex agglutination test. So we don't really have to remember all of it in detail, but remember that there are three types of demonstration we can use. A negative staining, McFadden capsule staining, where we are using this methylene blue stain, and third is serological test. In serological test, we can go for capsular serotype or we can go for antigen sample by using latex agglutination test. And if you wanna go really over it, then you can remember the name of the reactions. Now we are done with capsule. Let's move to the flagella. We'll be discussing about flagella and then we'll discuss spores and like that. So flagella are actually like appendages present on the bacterial cell wall, as you can see here. So some of the different types of bacteria with their different arrangement of flagella over them. Now we'll look into them one by one along with the examples. The first, as you can see here, there is a bacilli with one single flagella at the tip. This kind of flagellar arrangement is known as monotrichous. It is seen in case of Vibrio cholerae, Pseudomonas, and Campylobacter. Just try to remember one example at first, and you'll learn the others as we go on looking in the further chapters. Don't just pressurize yourself, okay? Now the second arrangement, we can see that there is tuft of flagella on just one end of the bacteria. This kind of arrangement is Lophotrichus, seen in case of Spirillum. The third arrangement is bacilli or bacteria with flagella all over the surface of the bacteria. Can you see it? So this kind of arrangement is known as peritrichous, seen in E. coli and S. typhi. The fourth and the last arrangement is amphitrichous, where amphi, it means two. So there are two flagella present at both ends. It is seen in case of Alkaligenes faecalis. So we saw four arrangement: monotrichous, one end single flagella; Lophotrichous, one end many flagella; peritrichous, all over the surface, many flagella; amphitrichous, two flagella, two ends. Just remember it this way. Now let's look into the structure of flagella. Flagella, it actually consists of three parts. They are basal body. The second one is the hook. And the third one is the filament. These three structures altogether, they make a flagella. As you can see, the basal body here, it is embedded inside the plasma membrane. then the periplasmic space, peptidoglycan layer, and all those. The hook, it is the part through which the filament arises. And filament here is the flagellin. It is made up of flagellin protein. Okay, the basal body, they are of different kinds. They can be different in structure in different bacteria. It is generally made up of ring-like structure, and this ring can be 2 to 4 in number. So this was about the structure of flagella. Just a basic idea about it. You don't really need to know much in detail. Now, for detection of flagella. Now, as we know that not all the bacteria they have flagella, some may be non-flagellate, and even if the ones which have flagella, they can be of different type. So through the detection of flagella, we can actually make out which bacteria it is that we are looking into. Okay, so the first method of detection is direct demonstration. We can do it we can do it by tannic acid technique or direct electron microscopy. The second method is the indirect method, where hanging drop method, Craigie tube method, semi-solid method, dark background or phase contrast microscopy method are included. So hanging drop method is a name where you're going to hear it many times ahead. Then there is dark background or phase contrast microscopy. This is a type of microscope under which you can demonstrate a flagella. Now let's look into the bacterial spores. You must have heard of spores. Spores are like the vegetative part of bacteria which grow in unfavorable condition. When the condition, they kind of not grow, actually they are produced, or the bacteria make it for to survive the unfavorable condition, and then they germinate when the condition reverses to a favorable one. So the bacterial spore, they are highly resistant resting stage of bacteria, or you can say it's a dormant stage, which is formed in unfavorable condition as a result of depletion of exogenous nutrition. When there is depletion of nutrition, there is no nutrition outside, and the conditions are unfavorable for the growth, the bacteria goes into this dormant state or resting state known as spore. The structure of spore is very simple one. The from innermost to outermost, it consists of core, cortex, coat, and exosporium. Here is a diagram for you people. So this is the innermost structure. We can see this is the DNA. Just right where the DNA is, it is known as the core. It is the innermost structure, the core. Then the second one here is the cortex. One layer outside it is the cortex. Outside the cortex, it is lined by outer membrane, and the core is lined by inner membrane. Okay. Then core is lined by germ cell wall and inner membrane. Outside the cortex is the coat. This is the coat. I'm sorry for the sloppy diagram, but you can make it out, I guess. And the outermost is the exosporium. So from inside to outside, we saw core, then there is cortex, there is coat and exosporium. Inside the core, you can find their DNA or the genetic material. Then there is core which is lined by germ cell wall. Then there is inner membrane, cortex lined by outer membrane, coat and exosporium. Now, what is sporulation? Sporulation is a process of formation of spore from the vegetative stage of bacteria. So it is not a method of reproduction, because bacteria is not actually dividing. The bacteria, it forms spore, but it is not dividing, that the number is not increasing. So it is not a method of reproduction. It commences when growth ceases due to lack of nutrition. It is actually a very complex process and takes about 10 hours in bacteria. Mature spore formed in extremely resistant the mature spore which are formed, they are extremely resistant to the heat and disinfectants. It is due to deposition of calcium and diplolinic acid into spore cortex. So we saw the cortex, right? It was the second layer from inside the cortex here. So in this cortex, there is deposition of calcium and dipicolinic acid. Calcium and dipicolinic acid, these are the two material which or the substances which make the bacterial spore extremely resistant. Please remember, it can be asked in MCQ. Now moving to the process of germination. The formation of dormant spore into active vegetative cell when grown in nutritive rich media is known as germination. The example of spore forming bacteria are Bacillus, Clostridium. You must have heard of Clostridium. Now the demonstration of spore, again we have seen the demonstration of capsule, we saw the demonstration of flagella. Now let's look into the demonstration of spores. It can be done by gram staining. In gram staining, they appear unstained refractile body within the cell. So supposedly a cell is there and it is all stained either purple or pink, but this one, these spores, they appear unstained and as a refractile body. The second method is modified Zell-Nielsen staining or modified Zn staining. The spores, they appear weakly acid fast, and special technique like Molar stain or Skaffer-Filton stain, this can be used. So this was all about spore. Now the bacteria, it actually produces two kind of toxins. We'll talk about toxins now. Endotoxin and exotoxin. The endotoxin and exotoxins, these are actually the main cause of or root of bacterial infection. Under the pathogenesis of bacterial infection, we'll read about these toxins. So first, let's talk about exotoxin. Exotoxin, do you remember the cell wall of gram-negative bacteria? There I had told you that the layer in the gram-negative bacteria, there was peptidoglycan, then there was lipopolysaccharide layer, and there was outer membrane. This lipopolysaccharide layer, it had three parts: lipidae, pore, and O protein or O antigen, O specific antigen, right? You remember that part? So that lipidae portion of lipopolysaccharide, it actually forms the endotoxin in bacteria. These are the integral part of cell wall of gram-negative bacteria, and they are released from bacterial surface by natural lysis of the bacteria. So bacteria does not secrete it, but bacteria actually undergoes the process of lysis or burst to produce this endotoxin. The various biochemical effects of this endotoxin are: first, macrophage activation. Please look into this part really carefully. Macrophage activation. The endotoxin, they bind to specific receptors which are present on macrophage, and they stimulate the release of acute phase cytokinin. These acute phase cytokinin include interleukin 6, interleukin 1, TNF alpha. These are the acute phase cytokine. Interleukin 6, interleukin 1, TNF alpha. So what happens is when this endotoxin binds to specific receptor present on macrophage, it causes release of these cytokines, that is interleukin 6, 1 and TNF alpha. These cytokines eventually cause fever, inflammation and activation of immune system. This was the first step or the first thing which the endotoxin does. Then The next step is complement activation. As the concentration of endotoxin rises, it activates the alternative pathway of complement. There are three different pathways of complement. One of them is alternative pathway. This alternative pathway causes release of C3A and C5A. This promotes the inflammatory cell chemotaxis. So now that the C3A, C5A are released, they call more and more inflammatory cells, which ultimately lead to high-grade fever, hypotension, shock by vasodilation, and capillary leakage. This was the process of complement activation. Apart from that, there is endothelial activation. Endothelial activation will lead to increase in vascular permeability. There, the permeability of blood vessels increases, and it can further lead to shock and all the condition, the extreme conditions. Then the fourth is coagulation pathway activation. The activation of Hagman factor and the other coagulation factor, it can lead to thrombosis and DIC. Fifth one is platelet activation. The endotoxin can cause release of mediator which cause increase in vascular permeability, thrombosis, DIC. The sixth one is mast cell activation. It can cause release of mediator like histamine which cause muscle contraction, increase vascular permeability. And the seventh one, in gram-negative septicemia, endotoxin are released in large quantity. They cause increased fever, petesia, DIC, which may result in shock and possibly death. So all these six steps we saw just now, they are the effects which endotoxin can exert on our body. First, they can lead to macrophage activation, causing like by binding on macrophage. They can cause release of acute phase cytokine, cause fever, inflammation, activation of immune system. Second, they will activate the complement pathway, activate the alternative pathway of complement, release C3, C5A, and promote inflammatory cell chemotaxis, leading to fever, hypotension, shock, capillary leakage. There can be endothelial activation, increasing its permeability. There can be coagulation pathway activation, leading to activation of Hagman factors, coagulation factors, causing thrombosis and DIC. It can lead to platelet activation, releasing mediators which cause increased permeability, thrombosis, DIC. The sixth is mast cell activation, release of mediators, histamine, muscle contraction, and vascular permeability. Gram-negative septicemia, it can ultimately result in shock and possibly death. So these are the effect that endotoxin can exert. So you can see it is quite dangerous for the host cell. Now the second toxin produced by bacteria are exotoxin. These are heat labile proteins, and they are present in both gram-negative and gram-positive. The endotoxins were specifically found in gram-negative bacteria, but these exotoxins are present in both gram-positive and negative. They diffuse readily into the surrounding. That means when the bacteria is alive only, these exotoxins are released into the surrounding, which is unlike the endotoxin, which are released by lysis. Now the exotoxins, it is also used for vaccine or process of making vaccines. It can be converted into toxoid by treatment with formaldehyde. It lacks the toxicity but it retains the antigenicity, thus it can induce protective immunity when used as a vaccine. Then the specific action, it is highly specific for a particular tissue like tetanus toxin for CNS. So these are the function or the uses of exotoxin. Now here is a table which will tell you about the difference between exotoxin and endotoxin. First, the nature of endotoxin is LPS, whereas exotoxin is protein in nature. The endotoxins, the main source is gram-negative bacteria. Exotoxin can be found in both. I told you endotoxin are released by lysis. They are not secreted. exotoxins are actively secreted. The endotoxins are heat stable, exotoxins are heat labile. The mode of action is, we saw so many, but among them, major one is the release of interleukin 1 and TNF alpha, that is the cytokine release, whereas exotoxin causes enzyme-like action. Effect of endotoxin can be fever, DIC. We saw so many like effects, and ultimately death and shock and death can also be one of the complication. But exotoxin effect is specific. Specific means if it depends on what kind of tissue or what tissue or organ these exotoxins are affecting. If it affects the CNS, it's gonna cause action which are very specific to CNS. If it's affecting the GIT, it will lead to GIT effects. Are you getting me? Now, tissue affinity. Endotoxin, they have no tissue affinity, whereas exotoxin are very specific about that. Fatal dose. Endotoxins are fatal in large dose, but exotoxin can be fatal in small dose. The endotoxin antigenicity is very poor, but exotoxin are highly antigenic. Endotoxins are not neutralized by antibody and they cannot be used for vaccine, but these features are present in exotoxin. So that's all about exotoxin and endotoxin. And yeah, this was all about the bacterial structure also. I highlighted the important points. I told you the questions which can be asked in theory and in viva also. These differences between exotoxin and endotoxin, difference between gram positive, gram negative bacterial cell wall, all these things. Then there's capsules, about spore, process of sporulation, germination. These things can be asked in viva and in your uni papers as well. Now let's move into bacterial infection. So we'll start with gram-negative bacilli infection. The first family is Enterobacterus. The members of gram-negative bacilli, they are non-fastidious, they are catalase positive, oxidase negative, glucose fermenter, and nitrate reducer. So these are the basic feature of this member of this family, Enterobacteriaceae bacteria. Most of them, they are commensal in human intestine, and they are known as coliform bacteria. Examples are E. coli, Klebsiella, Proteus. The enteric pathogen or the bacteria which can cause disease are Shigella and Salmonella. Based on lactose fermentation, these members are grouped into lactose fermenter and non-lactose fermenter. Remember, these example of lactose fermenter and non-lactose fermenter are asked very frequently in vivas. Lactose fermenters are E. coli and Klebsiella. Non-lactose fermenters are Shigella, Salmonella, and Proteus. Coming to E. coli are harmless commensal in human intestine. They are most common pathogen which are encountered clinically. They are associated with various clinical infection like UTI, then in uropathogenic E. coli, diarrhea like diarrheogenic E. coli, others like pyogenic infection, abdominal infection, pneumonia, and meningitis. E. coli can cause all of this. Lab diagnosis, we can actually culture E. coli on McConkey agar. The second one is Klebsiella pneumoniae. It can cause pneumonia. It is also one of the cause of UTI, meningitis, septicemia. It is lactose fermenter, non-motile, capsulated, and produce mucoid colony. The third one is Shigella. It can cause bacillary dysentery. It is one of the most important cause of bacillary dysentery. The four important species of Shigella are Shigella dysentery, flexneri, boydii, and sonnei. Lab diagnosis, the isolation of organism from the ideal stool specimen using enrichment media. The specific medium for Shigella are selenite F medium, and the selective medium like DCA, deoxycholate citrate agar can be used. The fourth bacteria here is Salmonella. It is divided into typhoidal Salmonella like S. typhi, paratyphi, type ABC, and non-typhoidal NTS. They colonize the intestine of several animals. So I'm just telling you about this organism in just and like not going in detail. This was the first family. The second one is Vibrio species. Then we'll talk about gram-negative bacilli. Actually, there are separate chapters on all of these most important uh, disease-causing bacteria. So you'll learn about them in detail in further chapters. But this is like just a very basic detail about them. So Vibrio species. Vibrio cholera, you must have heard of it, is the most important cause of cholera. Pathogenesis, it is due to potent enterotoxin, cholera toxin. The clinical manifestation, we can see paleness, watery diarrhea, rice like watery diarrhea. Then there is severe dehydration, renal failure, and then live diagnosis in TCBS, selective media can be done. Non-fermenting gram-negative bacteria include Pseudomonas aeruginosa and Acinetobacter. So this was all about this was all about general bacteriology. I know I haven't touched all the topics, but I did try to cover all the important points from the Apoorva's history book. If you have any confusion till now, please do let me know in the comment section. And if you want a video on any of the specific topic, any question answer related or anything, you can just let me know in the comment section. I'll surely try to look into it. Now, after this is the general virology. Virology is study of viruses. Here, two beautiful pictures of viruses drawn here. So the first one we can see is an envelope virus with icosahedral nucleocapsid. So this is a nucleocapsid which is icosahedral in structure, not so perfect, and this is the nucleic acid of the bacteria. Here we can see the lipid part of the envelope, and these are the protein part. It's a very simple structure, we can very easily remember this. This is the DNA or nucleic acid, can be RNA as well. This is the icosahedral nucleocapsid. Then this is the two parts. The envelope is made up of two parts. The inner one is lipid part and the outer one is protein part. See, then now here is the envelope virus with helical nucleocapsid. This is actually the half part, and imagine this is the covering and this virus is half opened, so it's drawn something like that. This is a helical, this kind of nucleocapsid is there, inside which the nucleic acid is present. These are the capsomers, and again here this is the protein and lipid part of the envelope. Now the important part in case of virology is different methods of viral cultivation. So I'll mainly look into this, and then we'll see the cytopathic effect, and that's it. This will be the last topic of the video. So quickly about the different methods of viral cultivation. The viruses, they cannot be grown on artificial culture medium. They are cultivated by animal inoculation, embryonated egg inoculation, or tissue culture. First, the animal inoculation. They are largely restricted for research purpose. Like in case of animal inoculation, as the name suggests, we inoculate viral particles or the whole virus in any animal and use it as a laboratory animal to see the or do the study purpose or for the study purpose. So this kind of inoculation is largely restricted for research purpose only. The second is the egg inoculation, where the embryonated hen's egg has been used, and this egg, it has 4 sites which are specific for growth of certain viruses. First is the yolk sac, second amniotic sac, then the allantoic sac and chorioallantoic sac. The yolk sac inoculation is used for arbovirus like rickettsia, chlamydia. Amniotic sac is used for inoculation of influenza virus. Allantoic sac is used for inoculation of viral vaccine, for the better yield of viral vaccine, and chorioallantoic is used for isolation of pox virus. The third method, the tissue culture method, it is of 3 types. It can be organ culture, implant culture, or it can be cell line culture, which is only method in use now, and this cell line culture is widely used, and you'll also find it in lab diagnosis of many of the viruses. So a little bit about cell line. It is of 3 types: primary, secondary and continuous. The primary cell line culture, some of the example are monkey kidney cell line, and then there is chick embryo cell line. Secondary or diploid cell line is human fibroblast cell, MRC5, WI38. Then there is continuous cell line like McCoy cell line, Vero cell line, BHK cell line. You'll find these cell lines further as you do study about the viruses in detail in their lab diagnosis. Now, what is cytopathic effect or CPE? It is a method which is used to detect the growth of virus in a cell culture. Supposedly we have done a cell culture of virus, now we want to see the growth of the virus. The method which we are going to use is cytopathic effect. It is defined as morphological change which is produced by the virus in the cell line detected by a light microscope. So the morphological changes produced in viral cell line which we can detect by a light microscope is cytopathic effect. The cytopathic virus, not all the viruses are cytopathic, but few viruses they can produce CPE, they are known as cytopathic viruses. Types of CPE are rapid crenation and degeneration of entire cell sheet seen by enterovirus. Second is Senetrium or multinucleated giant cell formation. It is seen in measles, RCV and HSV. Diffuse rounding or ballooning of cell line seen in HSV. Cytoplasmic vacuolation seen in SV40. Then there is large granular clumps resembling bunches of grapes seen in adenovirus. So just try to remember as you go on revising. I am sure you'll remember all of this by heart, but let me repeat it for you. Let's go through this once again. I know this is hard to remember. Rapid crenation or degeneration of the entire cell sheet. So what was this actually? CP, we are seeing the morphological changes. These are nothing but the morphological changes which are produced by the virus in the cell culture cell line. So the degeneration of entire sheet, if you see there is degeneration of entire sheet in the cell line, you can suspect that the virus is enterovirus. If there is syncytium or multinucleated giant cell formation is seen, multinucleated situation is seen, it can be the measles, RSV or HSV. If there is diffuse rounding and ballooning of the cell line, you can suspect it to be HSV. If there is cytoplasmic vacuolation, formation of vacuoles in cytoplasm, it can be SV40 virus. If there is large granular clumps or there is resembling bunches of grape-like formation is there, you can suspect it to be adenovirus. So this was all about cytopathic effect, the general virology and general bacteriology. With this, we'll be moving forward and I'll be bringing you more and more chapters of microbiology. Just stay tuned and please subscribe to the channel if you like my videos. Thank you for watching.